In this slide set, we will explore the molecular basis of genetic variation, including mechanisms and types of DNA sequence variations, the importance of DNA sequence variations, and the patterns of inheritance of genetic disorders. The image on this slide is a photograph of the model of the DNA molecule built by Drs. James Watson and Francis Crick in 1953. Drs. Watson and Crick used this model to depict their proposed structure for the DNA double helix. The hypothesized structure was derived from X-ray diffraction data produced by Drs. Maurice Wilkins and Rosalind Franklin. The model was constructed from metal scraps obtained from a machine shop. Drs. Watson and Crick published their DNA structure in the journal Nature on April 2, 1953. For their work, Drs. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962. Dr. Franklin died before 1962. Since Nobel Prizes are awarded only to living individuals, she could not be honored. A video entitled Exploring Our Molecular Selves provides a 3D computer-animated illustration of the basic principles of molecular biology, including transcription and translation. The video was produced by the National Human Genome Research Institute with co-sponsorship of the Office of Biological and Environmental Research, the U.S. Department of Energy, the Howard Hughes Medical Institute, the Pharmaceutical Research and Manufacturers of America, Nature Magazine, Science Magazine, and the American Society of Human Genetics. The video is available free through the website of the National Human Genome Research Institute. Human genetic disorders can be caused by a variety of mechanisms. Chromosomal disorders are caused by alterations in the number or gross structure of chromosomes. Examples of chromosomal disorders include Down syndrome and Turner syndrome. Contiguous gene deletion syndromes result from large, continuous deletions of the genetic material of a chromosome that cause several to many genes to be lost. Examples of contiguous gene deletion syndromes include DeGeorge syndrome and Williams syndrome. The signs and symptoms of genetic disorders caused by chromosomal abnormalities, including contiguous gene deletion syndromes, are extremely variable. In the case of structural rearrangements of chromosomes and contiguous gene deletion syndromes, most physical effects of the abnormality are likely due to disrupted expression or regulation of genes at or near the sites of the abnormality. In the case of chromosomal aneuploidies, alteration of the normal copy number of genes on the involved chromosome is likely the cause of most of the physical effects. Single gene disorders occur when a mutation is present in a single gene. The symptoms of the disorder are typically tied specifically to the function of the altered gene. Examples of single gene disorders include cystic fibrosis, Duchenne muscular dystrophy, Fragile X syndrome, and Huntington disease. Single gene disorders can be autosomal, that is, occurring due to mutations in genes located on chromosomes 1 through 22, X-linked, occurring due to mutations in genes located on the X chromosome, Y-linked, occurring due to mutations in genes located on the Y chromosome, or mitochondrial or maternally inherited, occurring due to mutations in genes located on the mitochondrial chromosome. Common complex disorders are disorders in which multiple genetic and or environmental factors act together to influence the expression and severity of a disorder. Common complex disorders include high blood pressure, obesity, and diabetes. The genetic location of the genes altered in association with a genetic disorder affects the pattern of inheritance of the disorder, that is, the way a disease is inherited in a family. The pedigree shown in this slide illustrates an autosomal dominant disorder caused by mutation of a single gene that is being passed from one generation to the next throughout a family. Knowing the pattern of inheritance of the disorder allows prediction of genetic risks for family members. Knowing the gene involved in the disorder allows genetic testing to identify individuals at risk for the disease or at risk for passing the disease gene to their children. In pedigree drawings, boxes are males, circles are females, shaded shapes are affected individuals, open shapes are unaffected individuals. The diamond at the bottom of the pedigree represents a fetus of unspecified gender. In the autosomal dominant pedigree drawn here, the fetus would be expected, prior to genetic testing, to have a 50% chance of inheriting the disease gene. If a genetic test were available for the disorder in question, the test could determine whether this fetus inherited the disease-associated copy of the gene or the normal copy of the gene from his or her affected mother. Mendelian disorders follow Mendel's rules of inheritance. They can be inherited in an autosomal dominant, autosomal recessive, X-linked, or Y-linked manner. Autosomal dominant inheritance is typically characterized by the appearance of a disorder in every generation of a family, passed down from parent to child for several generations. Autosomal dominant inheritance was sometimes described as vertical inheritance, although this term is not commonly used today. The word trait is typically used by geneticists to describe genetically inherited physical characteristics or features that are not considered to be diseases or disorders. Autosomal dominant traits are inherited in a manner similar to that of autosomal dominant disorders. Humans typically carry 2 copies of each autosomal chromosome, that is, chromosomes 1 through 22, and thereby 2 copies of each gene on those autosomal chromosomes. Most individuals affected by autosomal dominant disorders carry 1 affected and 1 normal copy of the gene associated with the disorder. These individuals are said to be heterozygous for the disease gene. Homozygous affected individuals carrying 2 affected copies of a gene associated with a disorder can occur, especially if the disorder is common. For some autosomal dominant disorders, homozygotes, that is, persons with 2 copies of an affected gene, are more severely affected than heterozygotes, persons with only 1 affected copy of the gene and 1 normal copy of the gene. Achondroplasia, the most common form of short-limb dwarfism, is an example of this phenomenon. In general, males and females affected by autosomal dominant disorders will be affected with equal frequency and severity. But there may be exceptions, such as cases where a particular disorder affects the genders differently. Male-to-male transmission of the disorder can occur in families with affected males. The children of affected heterozygous individuals typically have a 50% chance of inheriting the disease-associated gene and being affected, unless the disorder demonstrates reduced penetrance. Reduced penetrance is a phenomenon in which some gene carriers remain unaffected, despite the fact that they carry a gene associated with a disorder. One explanation for this phenomenon is that the gene is necessary but not sufficient to cause the disease. In these cases, the child has a 50% risk of inheriting the gene from an affected parent, but may not show the disorder. Some autosomal dominant disorders show reduced penetrance. Others do not. The degree of reduced penetrance associated with a particular disorder depends upon the disorder in question. Persons not affected by autosomal dominant disorders generally do not have affected children. However, there are exceptions, such as when a new mutation in a gene occurs at some point during transmission of the gene from parent to child. The child could be affected by the disorder while the parent remains unaffected. The percentage of cases of a particular disease that arise from new mutations varies from disorder to disorder. Autosomal dominant disorders also frequently show a high degree of variability. Variability means that individuals who are affected by the disorder can display a variety of symptoms and a range of the severity of the symptoms. A heterozygous affected parent has a 50% chance of passing the gene associated with the disease to each of his or her children But if the disorder is highly variable, it may be difficult to predict the severity to which a child will be affected. In most cases, variability is thought to represent the influence of other genetic and/or environmental factors on the expression of the disorder. Examples of autosomal dominant disorders include Marfan syndrome, neurofibromatosis, and Waardenburg syndrome. The drawing on this slide illustrates an autosomal dominant disorder. In this family, the disease is passed from a man to his son, and then from the son to his daughter. Individuals affected by autosomal recessive disorders carry 2 copies of the gene associated with the trait or disorder. Autosomal recessive inheritance is frequently observed as the appearance of a disorder in only one generation of a family, as an isolated case or as affected siblings and/or cousins. Autosomal recessive inheritance is sometimes described as horizontal inheritance, although this term is not commonly used today. In the case of common recessive disorders, or disorders that lead to non-random mating, such as deafness, which is common and frequently leads to deaf-by-deaf matings, a pseudo-dominant pattern of inheritance may be observed. For genetic counseling and risk assessment, it is important to recognize the possibility that pseudo-dominant inheritance might be occurring in a family. In general, males and females affected by autosomal recessive disorders will be affected with the same frequency and severity, but there may be exceptions in cases where a disorder affects the genders differently. In general, autosomal recessive disorders do not show reduced penetrance or the considerable variability that can be associated with autosomal dominant disorders. However, there are autosomal recessive disorders for which variability in the severity of symptoms among affected individuals is observed. Most often, children affected by autosomal recessive disorders are born to unaffected parents. However, for certain autosomal recessive disorders, there may be very mild or atypical manifestations of the disease in a carrier parent. Frequently, in autosomal recessive disorders, there is no family history of the disease and no reason to suspect that an affected child could be born. Carrier couples, that is, couples in which both partners are carriers of the same autosomal recessive disorder, have a 25% risk of an affected child with each pregnancy. Examples of autosomal recessive disorders include cystic fibrosis, Tay-Sachs disease, and sickle cell disease. Under what conditions widespread screening should be undertaken for the identification of unaffected carriers of autosomal recessive disorders is a topic of much debate. At present, most screening programs focus on high-risk groups, such as screening for cystic fibrosis carriers among Caucasians, and screening for cystic fibrosis, Tay-Sachs, and other genetic disease carriers among Ashkenazi Jews. The drawing in this slide illustrates an autosomal recessive disorder. In this case, the affected child is born to unaffected parents with no family history of the disease. X-linked dominant disorders are characterized by the appearance of a disease in both males and females in several generations of a family. True X-linked dominant disorders are few in number. There are many more X-linked recessive disorders. X-linked recessive disorders are typically observed as affected males born to unaffected parents. However, it may be more accurate to describe X-linked disorders simply as X-linked and not recessive or dominant, because with many disorders, historically described as X-linked recessive, carrier females can demonstrate variable degrees of signs or symptoms of the disease. For X-linked disorders, there can be a family history of the disorder in the form of affected uncle-nephew pairs, affected brothers, or affected male cousins, depending upon the carrier status of the females in the family. For common conditions, a pseudo-dominant pattern of inheritance observed as an affected male with an affected father can also be seen. However, in such cases, the affected son actually would have had to have inherited the trait from his mother, because males inherit their father's Y chromosome, not their father's X chromosome. It is important to understand the pattern of inheritance and genetic location of the associated gene, so that accurate risk analysis and genetic counseling can be performed for families. In the case of X-linked disorders, father-to-son transmission is never seen because fathers give sons their Y chromosome. Each son of a female carrier of an X-linked disorder has a 50% chance of being affected. Daughters have a 50% chance of being a carrier. All daughters of an affected male will be carriers because fathers have just one X chromosome to contribute to a daughter. Examples of X-linked disorders include Duchenne muscular dystrophy, hemophilia A, and hemophilia B. In addition, the classic red-green colorblindness trait is inherited in an X-linked recessive manner. Because it is so common, colorblind females who carry a copy of the gene associated with colorblindness on both of their X chromosomes are sometimes seen, as are colorblind father-son pairs. However, because we know the inheritance pattern of the trait, we know that a son inherits the trait from his mother, not his father. Roughly 8% of U.S. Caucasian males are colorblind, while less than 1% of females are colorblind. In the drawing on this slide, an uncle-nephew pair affected by an X-linked disorder is shown. In this case, the women in the pedigree with dots in the center of the circles representing them are obligate carriers of the disorder. The sister of the affected boy in the third generation of the pedigree has a 50% chance of being a carrier. Diseases of the mitochondria are inherited in one of two ways, through the maternal lineage or as autosomal disorders. Mitochondria, which are located inside the cell but outside the nucleus, carry their own small circular genome that encodes 13 proteins, 2 ribosomal RNAs, and 22 transfer RNAs. Mitochondria also import proteins encoded by nuclear genes. Diseases of the mitochondria caused by mutations in genes carried on the mitochondrial chromosome are inherited through the maternal lineage, passed from affected mothers to all of their children, but never from affected fathers, because only eggs contribute mitochondria to the embryo. This pattern of inheritance is illustrated in the slide. Diseases of the mitochondria caused by mutations in genes carried on the nuclear chromosomes demonstrate an autosomal pattern of inheritance that is dependent upon the gene or mutation involved. Individual mitochondria frequently carry multiple copies of their genomes. A single cell can carry many mitochondria. Not all of the mitochondrial genomes a person carries will necessarily carry a particular mutation. When all of the mitochondrial DNA present in an individual carries a particular mutation, that individual is said to be homoplasmic for the mutation. If some normal and some abnormal mitochondrial genomes are present in an individual, that person is said to be heteroplasmic for the mutation. Because of the complexity of the underlying genetics, maternally inherited mitochondrial disorders are remarkable for their variability. This variability is believed to be based on the relative percentage of mutant mitochondrial genes a person carries, the number of abnormal mitochondria that result, and the tissue distribution of those abnormal mitochondria. As a result, it can be difficult to predict the severity of maternally inherited mitochondrial disorders in the children of affected women. Examples of maternally inherited mitochondrial disorders include aminoglycoside-induced ototoxicity, which is a form of hearing loss, and mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, also known as MELOS. Multifactorial disorders, which include common complex disorders, are characterized by the interaction of multiple genetic and/or environmental factors that influence the expression of a disorder. Although common complex disorders may not show a clear pattern of inheritance like Mendelian disorders do, there is typically a familial aggregation of the disorder. That is, close relatives of an affected individual may be at increased risk for the disorder compared to the general population. Examples of multifactorial diseases include some forms of diabetes, high blood pressure, and obesity. Disorders associated with a new mutation occur when a mutation happens prior to or during gametogenesis or after fertilization. The mutation is found in the child, but not in either parent. Timing of occurrence of the mutation is important to the tissue distribution of the mutated gene, the symptomatic expression of the disease, the risk for additional affected offspring of the parents, and the risk for future offspring of the affected individual. For example, if a mutation occurs in the gonadal tissues of an individual with a new mutation, there is an increased risk to future offspring of that person. An advanced paternal age effect is characteristic of some new mutation genetic diseases. Examples of diseases with a high new mutation rate include achondroplasia, Marfan syndrome, Waardenburg syndrome, and neurofibromatosis. Note that these are all dominant disorders. Although recessive new mutations surely occur, it is far more likely that both parents of an affected individual are both carriers of the recessive mutations. Imprinting is a form of gene silencing whereby certain genes are methylated during gametogenesis to impart a parent of origin effect on gene expression. For example, if a gene is maternally imprinted, that is silenced in the maternal copy during female gametogenesis, only the paternal copy of the gene is expressed in the child. Paternal imprinting of genes also occurs. During gametogenesis, parental imprints are erased and reset in a manner consistent with whether the gametogenesis is occurring in a male or a female. A number of human genetic diseases occur because of mutations in imprinted genes or in the control sequences responsible for setting, resetting, or controlling imprinted regions. For example, if a person carries a mutation in an imprinted gene, the disease will be expressed only if the mutation is inherited from the parent whose gene is expressed. Imagine a mutation in a gene that is imprinted in the maternal genome. If a mutation in this gene is inherited from one's mother, the disease will not occur because it is the father's gene that is expressed. However, if the mutation is inherited from one's father, the disease will be evident. Another source of abnormalities in imprinted genes involves the imprinting centers that control the establishment of an imprint and the switching of imprints in gametogenesis. Mutations in imprinting centers can result in disease because of the failure to erase the grandparental imprint during gametogenesis. Examples of disorders associated with imprinted genes include Angelman syndrome and Prader-Willi syndrome. These disorders can also be caused when errors in meiosis result in a child who inherits both copies of his or her chromosome 15 from a single parent. This results in inheritance of only imprinted copies of the gene. This phenomenon is called uniparental disomy, or UPD. The appearance of a genetic disease at progressively earlier ages of onset and with increasing severity in successive generations of a family is called anticipation. The molecular genetic phenomenon behind anticipation is a length expansion of repetitive DNA sequences within a gene during transmission of the gene from generation to generation. These expansions cause increased severity of the disease with growing repeat length. Examples of diseases that show anticipation include myotonic dystrophy, Fragile X syndrome, and Huntington disease. There are two types of genetic heterogeneity: allelic and locus. Allelic heterogeneity occurs when different mutations in a single gene cause a particular disease. One disease that shows allelic heterogeneity is cystic fibrosis. More than 1,000 different mutations have been identified in the gene associated with cystic fibrosis. Understanding allelic heterogeneity is important for interpreting DNA test results and predicting disease severity. Locus heterogeneity occurs when a particular disorder is caused by mutations in more than one gene. Understanding locus heterogeneity is important for selecting the proper DNA tests to order for a patient interpreting DNA test results, predicting disease severity based on those DNA test results, and predicting disease risk for family members. X inactivation, sometimes called lionization, occurs during embryogenesis and is the process by which most genes on one of the X chromosomes of a female are inactivated. This mechanism provides dosage compensation and equates the level of gene expression of most X-linked genes in females to that of males who have only one X chromosome. There are some X-linked genes in females that escape X inactivation. X inactivation usually occurs randomly within the cells of a female, such that approximately half of the cells of a female express the genes from one of her X chromosomes, and the other half of her cells express the genes from her other X chromosome. However, skewing can occur in X inactivation. Skewed X inactivation is observed when one of the X chromosomes of a female is preferentially inactivated in substantially more than half of her cells. In some cases, skewing of X inactivation is extreme and may be caused by variations in the survival of cells in which a particular X chromosome is inactivated. Skewing of X inactivation can, in rare cases, cause X-linked diseases to be expressed in females. Mutations are changes in the DNA sequence of a gene. Because of the redundancy of the genetic code, mutations can, but may not necessarily, alter the sequence of the protein encoded by a gene. Several types of changes can occur in a DNA sequence. Some simple examples are shown in the slide. Substitution mutations are the replacement of one nucleotide for another. Deletions are the loss of one or more nucleotides. Insertions are the addition of one or more nucleotides. The inserted nucleotides can come from a variety of sources, including foreign DNA from viruses, transposable elements from other parts of the genome, or duplications of nearby or adjacent DNA sequence. Frame shift mutations alter the reading frame of a messenger RNA in such a way that the amino acid sequence downstream from the mutation is altered. Frame shift mutations are often caused by small insertions or deletions in genes and typically result in the appearance of a stop codon somewhere downstream from the mutation. Rearrangement mutations can be inversions or other complicated alterations of the normal sequence of a gene. Nonsense mutations are substitution mutations that result in the substitution of a stop codon into a protein at a position where an amino acid would normally occur. Nonsense mutations result in the premature truncation of a protein. If the nonsense mutation occurs at the beginning of the gene, it may result in a complete loss of the functional capabilities provided by a protein derived from the mutated gene. If it occurs near the middle or end of the gene, it may result in the production of a protein missing only some of its functional capabilities. Missense mutations result in the substitution of one amino acid for another in the sequence of a protein. Missense mutations can interrupt the functional capabilities of proteins in which they occur, or they may have no effect at all on a protein's function. Missense mutations that do not have pathological consequences can become frequent in the population because they are not selected against. Such mutations are typically called polymorphisms to distinguish their benign nature from that of pathologically significant mutations. Null mutations are mutations that result in the absence of a protein product from a gene. Null mutations can be nonsense or frame shift mutations if these mutations occur very early in the coding sequence of a gene. Functionally, null mutations can also be mutations that occur in the promoter regions of genes and eliminate expression of the gene. The human genome also contains a number of repetitive elements, or tandemly repeated sequences. These repeated sequences are spread throughout the genome and can vary in size from a few dozen base pairs in length to hundreds of base pairs in length. Some repetitive elements occur outside the coding sequences of genes. Others occur within the coding sequences of genes. The consequences of variability in the length of repetitive sequences depends upon their genetic location. Mutations can occur at any point in a gene and may have a variety of effects on a gene, including affecting its expression, the processing or stability of the messenger RNA produced, or the function of the encoded protein. Loss of function mutations result in the loss of some or all of the functional capabilities of the protein derived from the gene in which the mutation occurs. Gain of function mutations result in a protein that has acquired a new function that it did not have prior to the mutation. Sometimes the new function is harmful and can cause disease by disrupting cellular processes. Dominant negative mutations result in a protein that interferes with the normal functioning of other proteins. For example, if a protein acts as part of a multi-protein complex, a dominant negative mutation in one protein of the complex could interfere with the functioning of the entire complex. Alternatively, a null mutation may be less harmful because although there is less of the protein complex in cells, what is there functions normally. An example of such a disorder is osteogenesis imperfecta, which is caused by mutations in type 1 collagen genes. Null mutations typically result in less severe disease than dominant negative mutations, because although there is less collagen made from null mutations in individuals who carry a null mutation, what is made is normal. Dominant negative mutations interfere with the proper formation of collagen fibers and can cause much more severe consequences for a cell because all or most of the collagen made is abnormal. A polymorphism is a DNA sequence variation that is common in the population, as opposed to a mutation which is more rare. The term polymorphism also is used frequently in clinical genetics to distinguish a DNA sequence variation that is unrelated to disease from a DNA sequence variation that is associated with a disease. A polymorphism occurs in the human genome roughly every 250 to 1,000 base pairs. Polymorphisms provide markers or signposts throughout the genome that can be used by scientists to trace traits and diseases and their associated genes through families or large populations. This experimental approach has allowed researchers to map the genetic locations of numerous disease genes, facilitating their identification. The polymorphism itself may not necessarily be associated with the disease in question, but it may sit on the chromosome very near the genetic alteration associated with the disease, and thus may be used as a tag for the disease gene until a more precise genetic alteration associated with the disease can be identified. The slide shows an electropherogram generated by automated fluorescent DNA sequencing of a segment of DNA. The arrow shows a nucleotide substitution in one of the two copies of this gene in this individual. One copy of the gene carries an A nucleotide, indicated by the green peak, while the other allele carries a C nucleotide, indicated by the blue peak. This nucleotide substitution is detected as a dual peak in the output of the sequence analysis software. In this case, the DNA sequence variation results in a missense mutation. The detection of variations in the DNA sequence of a gene that are of uncertain biological consequence can complicate the interpretation of genetic test results. As we have already discussed, the human genome also contains a number of repetitive elements or tandemly repeated sequences. The size of these repeated sequences often varies among individuals. Many of these variable length repetitive sequences are benign and simply represent normal variability in the human genome, while others are associated with disease. The polymorphic nature of repetitive DNA sequences is the basis for much of the DNA-based forensic analyses performed today, including paternity testing and personal identification testing. The most common causes of mutations in DNA are errors during DNA replication and exposure to mutagens. Although the proofreading capability of human DNA polymerase reduces the error rate of DNA replication to less than 1 base per billion base pairs replicated, mutations can remain after DNA replication. Slippage of the DNA polymerase along the DNA strand during replication also can lead to small insertions or deletions within the sequence of a gene. Typically, slippage occurs in repetitive sequences, but conformational irregularities of the unwound DNA also have been shown to contribute to the occurrence of small insertion, duplication, or deletion mutations. Exposure to ultraviolet light, mutagenic chemicals, and radioactivity can also damage DNA. The damage caused and the consequences of the damage are related to the agent to which one is exposed, the age at which the exposure occurs, the dose and duration of the exposure, and the genes affected. For example, exposure of fetuses to large doses of particular mutagens may have more significant clinical consequences than short-term, low-dose exposure of an adult. Certain mutagens cause characteristic types of DNA damage. For example, radioactivity typically causes double-strand breaks in the DNA, while certain chemicals are more likely to cause nucleotide substitutions. Cells have built-in mechanisms for DNA repair. The first step in DNA repair is recognition of the DNA damage. The next steps depend upon the type of damage that has occurred. For example, if the damage is to a single nucleotide in a gene, typically the damaged base is removed and replaced through a process called nucleotide excision repair. Cells have specialized proteins that perform these DNA repair functions. Mutations in the genes that encode DNA repair functions can be the cause of genetic disorders. One example is hereditary non-polyposis colon cancer, or HNPCC, which results from mutations in proteins that repair mismatched bases in the DNA double helix. DNA sequence variations are a normal part of the genetic composition of every living thing. Only identical twins share identical genomic DNA. But even identical twins can have differences in their DNA if sporadic mutations occur after twinning. Changes in DNA are the basis for the adaptation, evolution, and speciation of living things. Variations in DNA are also the basis for susceptibility or resistance to genetically influenced disorders. DNA sequence variations are also a fossil record of the human species. Humans are more than 99.9% identical at the DNA level. However, with 3 billion base pairs in our haploid genome, this leaves a lot of room for variation. Thus, each of us is unique, with the exception of identical twins who share the same genetic makeup. Human genetic polymorphisms have been used recently to trace the history of our species. What has been learned is that humans evolved in Africa about 100,000 years ago. Although the dates are only broad estimations based on calculated mutation rates, we now know that we all are descended from a small group of perhaps 10,000 to 80,000 individuals. Over time, humans migrated out of Africa in several waves and eventually populated the globe. From these studies, we have learned that every human alive today is related to every other. We are all descended from the same trunk of the human family tree, brothers and sisters separated only by time and geography. In summary, DNA sequence variation is a normal part of every species and is not always associated with disease. DNA sequence variations provide a mechanism for mapping genes and tracing the genetic history of organisms. Understanding the genetic